Can you guys see it? Okay. So today we are going to design guide RNA for a knockout experiment. So you are going to basically try to knock out GAPDH. So what I have told you for the past 2 days, that for knockout, which exon you should target? For knockout, you should target the initial coding exon just after the ATG. Okay. So only then, if there is a indel formation here, then there will be a frame shift. So once there is a frame shift, due to the whole sequence getting changed, getting frame shifted, it will either code for some random protein or it will not code for any protein. Alright, so the gene will become silenced. So here, which is the exon that is the initial coding exon? Obviously this exon 2, as it contains the ATG followed by this. But one more thing you should also keep in mind, that you might not get proper guide RNA in this small sequence. Alright, because after ATG, just this region itself is 26 bases. You can see it here, how many bases that you have selected. And your guide RNA sequence is 20 bases. So what you can do is, you can make yourself more flexible. So this is exon 2. You can also target guide RNA for exon 3, because this is the next available guide RNA. Sorry, the next available exon. And if you don't find it here also, because there are a total of 9 exons, so you can design a guide RNA with exon 2, 3 and 4 in your design parameter. Okay, Monica. So Monica, do you see my screen? Hello, Monica. You can speak. You can turn on your microphone and speak.
Yes, sir.
So here you can see that exon 2 contains ATG, correct?
Yes, sir.
So after this, the protein sequence will start, like after mRNA is formed, correct?
Yes.
Yeah. So as I had showed in my PPT, that for knockout, you should target the initial exon just after the ATG.
Yes.
So what is the initial exon here?
Yes, sir.
What is the initial exon?
I don't know, sir.
Okay. So exon 2 has ATG. So obviously you will target exon 2. Okay.
Initially, the ATG, no?
Just after the ATG.
Yes.
Yeah, but this exon 2 is very small. You will not get much guide RNA sequences from this 26 bases sequence. So you can
excuse me, Sanket.
Yes.
I think Monica is facing the language problem. You can speak in Hindi, no problem. I think everyone knows the Hindi. I think Monica is facing the language problem. So
Yes,
maybe you can speak in Hindi. I think everyone knows the Hindi.
I can surely speak in Hindi, but there are people from southern part of India who might not understand. So I was concerned about
समझ में अभी आपने जो बोला ना मतलब स्नैप जीन पे पीपीटी देखने के लिए, वो नहीं समझ में, बाकी तो समझ में आ रहा था.
अच्छा, ओके. ठीक है. Okay, I'll just explain Hindi then. Right. So यहाँ पे तुम देख रहे हो कि exon 2 है, so इधर ATG सीक्वेंस है. So ATG सीक्वेंस के बाद ही क्या? लाइक तुमको क्या समझ में नहीं आ रहा?
अभी आपने स्नेप जीन पे बोला ना, उसके लेंगे वो चीज नहीं समझ में आया कि स्नेप जीन पे करना क्या है।
स्नैपजीन में तुमको विजुअलाइज करना है।
तो ये चीज नहीं समझ में कि करना कहाँ से है।
सो मैंने तुमको एक फाइल भेजा है व्हाट्सएप में, 2 फाइल्स।
यस सर।
वो डाउनलोड कर लिया है तुमने?
ह्यूमन जीन पीडीएच का।
हाँ, या डीएच डीएनए एंड एमआरएनए, 2 फाइल्स भेजा है।
यस यस
आपने डाउनलोड कर लिया है?
यस सर।
हाँ, उसके बाद मैंने एक और चीज़ भी भेजा था, लाइक ग्रुप में शायद कोऑर्डिनेटर ने भेजा था, स्नैपजीन कहाँ से डाउनलोड करना है, स्नैपजीन व्यूअर।
यस।
आपने वो इंस्टॉल किया?
स्नैपजीन ऐप नहीं इंस्टॉल हुआ था।
वो एक सॉफ्टवेयर है जो आपको लैपटॉप में
हाँ
इंस्टॉल करना है। आपने किया था वो?
यस सर।
हाँ, तो उसको आप खोलिए, स्नैपजीन खोलिए। आप अगर स्नैपजीन खोलेंगे तो आपको
आई एम नो यूज लैपटॉप, मैं जो भी कर रही हूँ फोन में ही कर रही हूँ ना।
यू डोंट हैव अ लैपटॉप?
यस सर। नो सर।
ओके,
आई यूज फोन ही।
मैंने मेंशन किया था कि लैपटॉप चाहिए।
यस सर, आपने मेंशन किया था, बट इस टाइम में जहाँ हूँ वहाँ हो नहीं पाया वो चीज़।
सो देन इट विल बी हार्ड।
यस सर।
मुश्किल होगा।
मोनिका,
तो आप
मोनिका एक काम कीजिए,
Yes,
आप अभी है ना ऑब्जर्व कर लीजिए, ठीक है। अभी ये चीज़ जो होती हैं आपका ये लैपटॉप पे होता है या कोई आपका डेस्कटॉप पे होता है, तो आप अभी इसे ऑब्जर्व
sir.
कीजिए। जब सर आपका कंप्लीट होने के बाद इसके पीडीएफ वगैरह भेजेंगे और जब आपके पास डेस्कटॉप अवेलेबल होगा तो उसको आप प्रैक्टिस कीजिएगा, परफॉर्म कीजिएगा। फ़ोन में एक्चुअली नहीं हो पाएगा अच्छे तरीके से, ठीक है।
हाँ मतलब लैपटॉप में फिर हम बाद में कर सकते हैं ना ये चीज़?
हाँ बिल्कुल बिल्कुल, संकेत सर का नंबर।
मैं जहाँ हूँ वहाँ नहीं है, बाकी है, लेकिन अभी मैं
हाँ वही मैं बोल रहा हूँ कि जब आपके पास डेस्कटॉप अवेलेबल होगा तो आराम से आप उसे परफॉर्म कर सकते हैं। आपको संकेत सर कोऑर्डिनेट कर देंगे, ठीक है।
ओके सर, थैंक यू सर।
ओके।
एंड मोनिका, मैंने वो अपना लिंक्डइन एंड जीमेल आईडी सब शेयर किया।
हाँ सर।
सो यू कैन कांटेक्ट मी एनी टाइम।
ओके सर।
या अभी के लिए ऑब्जर्व कर लो।
ओके सर, थैंक यू सर।
Yeah. So we have to design guide RNA for exon 2, 3 and 4. Why we are choosing exon 2, 3 and 4 is because exon 2 in itself is very small, because ATG sequence is coming in the middle, and in order to make a frame shift we have to target sequences that are after this ATG sequence. Alright, because targeting before the ATG sequence will not cause any benefit for us, because it's just a regulatory exon part. So this sequence in itself is just 26 bases. Okay, so we will take exon 2, exon 3 and exon 4 for our guide RNA design. Alright, so what you should do is you can open Microsoft Word. Okay, just open Microsoft Word, just copy, just click here. You can click this feature exon 2. Here you have an option called copying. Copy the top strand bases. Okay, and then you open your MS Word. Okay, and you paste it. This is exon 2. You can color code it. Okay, so let's say I have color coded exon 2 as red for your convenience. आपको जो कलर पसंद हो आप वो कलर लगा सकते हो, जस्ट आपके कन्वीनियंस के लिए. Then you copy exon 3 the same way, and then you paste it. Just put it here. इसको आप कलर कर लो कुछ और दो. साथ में लगा दो. Then you have exon 4. Copy it and then repeat the same thing. You can color this or something else. अभी ये जो टोटल सीक्वेंस है इसमें exon 2, exon 3 and exon 4. Yeah, sure, I'll repeat it. We have to basically copy exon 2. If you just click on this feature called exon 2, it is selecting the whole exon 2. Once it is selected, you can see it is becoming blue, which means it is selected. You have an option here, top left, called copy. Click on copy and then copy top strand bases. Alright, or Control C, whatever is convenient for you. Copy it and then paste it in Word. So your exon 2, you can put it as red color. And then same way you can copy exon 3, which is here, and exon 4. Again, why we are choosing exon 2, 3 and 4 and not just 2? Because our exon 2 is very small. Okay, so we won't get much guide RNA options from just designing guide RNA for this sequence alone. We will not get much guide RNA. We need more options. Once we have more options, we can choose the best ones, and then from that you will be ordering 3-4 sequences which you will validate in your lab. Alright, so for that we need more options. So in order to get more options, we are choosing exon 2, exon 3 and exon 4. Once copied, once you have put it in the Word, just copy the whole thing. Copied. Now what you should do is open your Google Chrome or whatever you have. Open chop. It will be mentioned. I will send this link in WhatsApp. You guys must have received the link. Please do click on it and open it. Can you guys open it? It will open something like this that you have here. Are you guys able to open? Okay, Savan got it.
Please respond everyone. You can erase your hand or you can respond through emojis.
Yeah. If anybody is facing any issue, please just unmute yourself and ask. Okay. I think almost everybody got it. So here you have your target. It is mentioned here. You have your this rich genome using CRISPR Cas9, obviously. As I had told you in my previous classes, there are other types of CRISPR Cas also, Cas12, Cas13. So we can maybe conduct another workshop some other day, but you can go and read about it, about what is Cas12 and what is Cas13. So they are for different purposes, and it is also interesting. And as I told you, for base editing and prime editing, there is Cas9 nickase also here. So you can explore these things on your own after today's class. Okay, and you have an option here for knockout, for knock-in, for activation, for repression, for nanopore enrichment. Just forget about nanopore enrichment. Okay, that's nothing. Activation, repression, as I taught you for base editing, prime editing, that thing is here. CRISPR A, CRISPR I. Today we are going to see about knockout. Okay, we are designing a guide RNA for knockout. So what you will do, first you will paste your target. Okay, so here you have to click on paste target. You can see here there is an option called paste target. So here you will open your Word file, copy this whole sequence that you have made, which contains exon 2, exon 3 and exon 4. Alright, and paste it here. Okay, that's simple. Everybody do it. Is everybody able to do it? Okay. Now, after pasting the target, okay, you have to choose which organism it is. Which gap pH is ours? Which organism's gap pH is ours? It is humans. Alright. So, here you have to check Homo sapiens. Okay, there are multiple options of Homo sapiens. You can choose the best one. Let's say we will choose the human genome 38. Okay, choose human genome 38, HG38, Homo sapiens HG38. Is everybody able to choose it? Okay, great. So obviously we are using CRISPR Cas9, okay, for knockout. So choose CRISPR Cas9 here, and in for section use knockout. Ultimately your selection should be something like this. The target must be pasted here, Homo sapiens, CRISPR Cas9 for knockout. Okay, everybody done it? Have done it? Okay, great. Okay. Next, there is something called options here. Okay, everybody can see there is something called options. Click on options. You have multiple settings available here. Alright, don't get scared about when you see about this settings. After today's class you will get some idea, and based on that you guys can go and toggle what other functions, what are these other functions for and how do they work. You can try it on your own. It will be very much interesting. So today we have copied and pasted the exon parts. Exon part means it is the coding region. Okay, so it is already on coding region, so basically you can click on coding region. Okay, it is not all exons including UTRs, splice sites, 5' UTR, 3' UTR, promoter. It's not that. It is only coding region. Okay, after that restrict targeting. So in this, what is mentioned here? Search exons and the immediate short flanking regions. Only search within the exon. Okay, so we want to increase our option. So what we can do is we can select search exons and the immediate short flanking regions. What do they mean by this term? Search exons and immediate short flanking regions. So let me just show you in Snapchat. Imagine this is an exon. Okay, if you choose this option, only search within exon, it will design a guide RNA whose all 20 of the 20 nucleotides will be inside the exon itself. Okay, it will be only inside the exon. It won't be outside, it won't be falling outside the exon. It will be only all the 20 bases, you can see here. All the 20 bases will be inside the exon only. Okay, understanding it? Whereas if you choose this option, search exons and the immediate short flanking regions, which means the NGG will be here. You can see here there is a NGG, TGG. But the 20 nucleotide PAM sequence will contain region of both the exon as well as the neighboring intron as well. So if we choose this option, it will obviously come and cut here inside the exon only. But the guide RNA binding will bind in between both exon as well as intron. So why are we choosing this? Because we will get more options if we choose this option rather than choosing just exons. Instead of choosing this option, we are going to choose this option. Okay, so this will give us more options. I hope you are understanding it. If anybody has any doubts, please raise your hand. Okay. Nobody Somebody has sent it out. Okay. Next is isoform consensus. As I had said yesterday, that one gene can contain multiple isoforms, and we should design a guide RNA which should go and bind, which is common, which region is common among all the isoforms. If you see my last PPT, I will show you. Yeah, here, this last point. Make sure the guide RNA targets an exon shared by the transcript isoforms you want to affect, which means you should design the guide RNA in such a way that that region is common among all the isoforms. It is not just specific to one type of isoform. Understanding? So, what should you choose here? Search all exons in all isoforms, or only search regions present in all isoforms. So it is intersection. Alright, we are searching regions that are present in all isoforms. So this is the option. It is already inbuilt, it is chosen, but this is why you are choosing this. Okay, whereas union means it will search all exons in all isoforms. Okay, which means it can, like the guide RNA that it will suggest, it might cut in one isoform, it might not cut in another isoform. But this one, it will, if whatever guide RNA is being suggested, it will go and cut in all the isoforms. This GC content is 20 to 80. It has this constant, like they have already inbuilt, set it at 20 to 80, but ideally it should be 40 to 60. Alright. Self-complementarity has to be below -1. What if the self-complementarity is more? The guide RNA will bind to itself. Because there are multiple guide RNAs produced, right? Wait a minute. We can see here, this guide RNA is binding to the DNA. But if the self-complementarity is more than -1, then this my guide RNA will go and bind with another same copy of guide RNA, or to itself alone, to this guide RNA scaffold or somewhere else. So this will reduce the efficiency of the guide RNA going and binding to the target site. That is why your self-complementarity has to be below -1. Okay, this restriction enzymes, you can ignore it because it will vary according to company. Like they are giving you an option. So once you understand something from this today's workshop, you can go and toggle and check what is the difference between this company's Cas9 with regard to Takara company's Cas9. What is the difference? You can find it. Okay, minimum size of restriction by the binding site, this is constant 4. Okay,